Hello everyone, this is Shraddha and you're watching Knowledge Center. In this video, we have going to discuss about the mechanism of the urine formation. So before watching this video, I recommended you people to watch my previous lecture so that you can better understand this mechanism, because in the previous lecture on the structure of the nephron. So once you watch that video, so you can better understand the concept of the structure of the nephron, then you can understand the mechanism of the urine formation, because the urine formation will be done in the nephron only. So link will be provided there in the description box, so you can check it out please. So mechanism will be done in the three stages of the urine formation. First is a glomerulus filtration, second is a tubular reabsorption, and third one is a tubular secretion. So here I wrote about the physiology of the urine formation. First is the introduction, second is the glomerulus filtration, third is the tubular reabsorption. In the site 1, it is done on the PCT only, proximal convoluted tubule. Site 2, it is done only loop of the Henle, and third is a site 3, it's done on the DCT, distal convoluted tubule. Fourth is a tubular secretion, last one is a conclusion. So this is my handwritten notes. I will explain after the explanation through the diagram. So urine formation is very important of our body, because urine is a waste product of our body. We have to excrete from our body, and we have excretory system like kidney, and we have two kidney in our body. This is very thankful to God. So urine formation is very important process, and urine is the end product of the metabolic mechanism, and this is very important function done by the kidney. So when we talk about the urine formation, so there is a three stages we have to read in this lecture. So we have blood, and blood is circulated all around our body. So from the afferent arteriole, it comes in the kidney, in the nephron. In the kidney there is a millions of nephron are present. So I just draw one nephron over here so you can better understand. So this is afferent arteriole and this is efferent arteriole. Blood comes through the afferent arteriole and goes through the efferent arteriole, means and you can see the afferent arteriole is a very wide and efferent arteriole is very narrow. Afferent arteriole comes and put the blood into glomerulus. This is glomerulus and this is Bowman capsule, and this is PCT, and this is loop of Henle, this is DCT, and this is collecting duct. Okay, so when blood comes through the afferent arteriole, so a large amount of blood comes in the glomerulus, but you can see that not equal amount of blood goes with the efferent arteriole, because of it is a very narrow in the size. So here the filtration have to be done in the glomerulus. Glomerulus is it is a filtrate, and it is just a network of the capillaries. Yeah. And what is capillary? We have three tubes in our body, was artery, vein, and the capillaries. Artery and vein don't have pores, means they are not absorb and secrete anything from the blood, but capillaries have little pores on over here, so they can secrete and absorb there something. So from the vein the blood comes through in the glomerulus, so it will filtrate in the Bowman capsule. A large amount of blood comes in the Bowman capsule, so here the blood can be filtrate. Total 70% blood filtrate over here in the first stage is glomerulus filtration. So the particular filtration is called the glomerulus filtration, and here what will happen, it is called a primary urine. Okay, and it contains water, glucose, amino acid, urea, uric acid, ions, means all the waste product from the blood. It can be absorbed over here along with the water, because it is permeable for the water and salt, both of it. So this filtrate leave Bowman capsule and it enter in the PCT. So this filtrate is called the glomerulus filtrate. So here the first process going to be complete, that is called glomerulus filtration, means blood filtrate in the tubules. So second one is the tubular reabsorption, and third one is the tubular secretion. So they both are done in the tubules. Which tubules? First is this one, vasa recta. Vasa recta is just a efferent arteriole goes forward and make a vasa recta. This is a blood capillaries, and this PCT, proximal convoluted tubule. It is going to be add with the loop of Henle. This is descending loop of Henle. This is ascending loop of Henle. It attach with the DCT, distal convoluted tubule, and DCT now attach with the collecting duct, and this duct is forming the urine drops. Okay, and this is a vasa recta. Vasa recta, this is ascending loop of the vasa recta, and this is descending loop of the vasa recta. And you know, the kidney is going to be 180 liter blood filtrate in the per day, and we just excrete 1.5 liter of urine in our body. So the next step is reabsorption of the 180 liter filtrate. Only 1.5 liter convert into the urine, while the remaining molecule and ions and water reabsorbed by the peritubular capillaries or vasa recta. So this is called the reabsorption by the tubules. So when the filtrate, glomerulus filtrate come in the PCT, so PCT is just permeable for the water and salt, both of it. So from the PCT, water will be absorbed from the vasa recta, sodium will be absorbed from the vasa recta, glucose will be absorbed from the vasa recta, and amino acid and absorbed from the vasa recta. And in PCT there is a co-transporter are present. Co-transporter means when sodium going to be transferred to the vasa recta, so it club with the glucose and make a sodium glucose co-transporter. Okay, and like this sodium amino acid transporter, sodium inorganic phosphate co-transporter are here present, and H2O means water. Water also transfer from here with the osmosis. Osmosis means a passive process. There is no need to use the energy. This is called a passive process. These all about ions absorbed by the vasa recta or peritubular capillaries. Also H2O absorbed by it. Okay, so this is called the reabsorption by the PCT, and approximate 100% water absorbed by the PCT. Now this filtrate comes in the loop of Henle. This is the loop of Henle, this is descending loop, and this is ascending loop of Henle. So filtrate when come in the descending loop, so the difference basically, the descending loop is permeable for the water and ascending loop is permeable for the salt. So all the water absorbed from the descending loop. Okay, so the filtrate will be concentrated because of salt. Here salt will increases the quantity and water will be decreases because water is absorbed from the vasa recta. So this filtrate going to be enter in the ascending loop of Henle. So there concentrate filtrate are entered in the ascending loop of Henle, and here salt will be absorbed from the vasa recta. So salt will be reabsorbed, and so the concentration of the filtrate is going to be less in this loop. Now the filtrate is going to be come in the DCT, means distal convoluted tubule. Okay, so here it is permeable for water and salt, both of it. So here just reabsorb remaining water and remaining salt and other ion from the tubules, means this tubule have formed the urine, so it don't have the important ion which are important for our body, so we have to reabsorb it. So this process is called tubular reabsorption. Okay, now this filtrate enter to the collecting tubule. So this second process going to be complete. Third process is called a tubular secretion. What is tubular secretion? So when the blood is just filtrate in the glomerulus, so when filtered blood comes in the efferent arteriole, but it is not completely filtered. It has some ammonia and some amino acids, some drugs are present which is not beneficial for our body, so we have to secrete it from there, otherwise they are circulating all around of our body. So this process is called tubular secretion, means tubular secretion done by the vasa recta. Okay, so this vasa recta just when it going to be enter in the loop of Henle. So it just secrete all excess drug, hydrogen ion, potassium ion, ammonia from this tubule, this peritubular capillaries or vasa recta, so that the blood going to be completely filtered. Okay, so it is comes to the this tubules, DCT, loop of Henle, and there. So now this blood is a just a filter blood, and this filtrate is going to be converted in the urine in the collecting duct, and then we just excrete from our body. So understand what is the basic process of the urine formation. First is the glomerulus filtration. It is done in the glomerulus only, or and filtrate comes in the loop of Henle, PCT. And second was the tubular reabsorption. So tubular reabsorption done in the loop of Henle. Okay, and third one is a tubular secretion. It is done in the vasa recta, means blood capillaries. These three stages are involved in the urine formation, and finally we just formate the urine and excrete out from our body. So there is my notes. So this is all about the physiology of the urine formation. This is the steps, this is the figure I have to make. Now in the introduction, there's a urine formation is very important for our body, and urine is the end product of the metabolic mechanism, and this is very important function done by the kidney. And physiology of the urine formation, this is all about the glomerulus filtration, means kidneys is a body's blood filtrate system. Blood enter in the glomerulus membrane. Here the tiny capillary are present, and glomerulus nested in the region called woman capsule. You can read here. And now it is comes into the tubular reabsorption. This is the next step of the tubular reabsorption, and this is site of PCT. There is a co-transporter present, means sodium glucose co-transporter, sodium amino acid co-transporter here. Okay, and they all are absorbed in the peritubular capillaries. Now it enter to the loop of Henle. Here descending loop of Henle, which is permeable for the water, not for the salt. Ascending loop, it is permeable for the salt, not for the water, and co-transporter are present like sodium, potassium, and chloride co-transporter are present, and hormone. Here the two hormone are excreted, ADH and aldosterone. Yeah. Now DCT, it is this. And now tubular secretion, some substance are not filtered by the Bowman capsule, so they are secrete from the tubular. So PCT and DCT, they are secrete. Now blood enter in the Bowman capsule and get filtered to, and filtrate comes in the glomerulus filtrate. Now low molecular weight filtrate, PCT, descending loop, ascending loop, then DCT, and collecting tubule, then form the urine. So this is all about the formation of the urine. Thank you so much for watching.